All right, welcome back. We are going to do a real statistical inference problem. We're going to try to calculate the probability of something causing cancer. So here's the background. We've got animals that were dosed with ethyl bad stuff. We want to know, does ethyl bad stuff cause an increase in the cancer rate? And we've got animals that were dosed with vehicle, right? Because in order to know if ethyl bad stuff actually did something, we need to compare it to some control. And the control is going to be the vehicle because we want to make sure that the vehicle isn't causing, or actually we want to be able to differentiate what ethyl bad stuff does from its vehicle. That's probably the easier way to say it, right? So the objective. What do we want to know? Does ethyl bad stuff increase the rate of cancer? That's what we care about. But like I said, in order to get there, we need to know what is the cancer rate in the vehicles. So here again, here are the steps in the Bayesian analysis. We got step one, we visualize the data. This data is pretty easy. There isn't much to visualize, so we can really just skip to building some models, which is step two. After we get these models built, then we got to validate them against the data. We're going to do that. Then we're going to make some inferences, and we'll do that too. So we're at step two. We're going to build some models, because like I said, we can't really visualize this. So we're going to ask some questions about this data. So the first one is there any historical or any other data that's going to help us? The next question is, what type of data do we have? Do we have count data? Count data is like, literally how many animals got cancer or tumors. Continuous data, that's a little different. That's going to be things like, when you do an RT-PCR or a gene expression assay, or you're measuring enzyme levels or maybe even enzyme kinetics. These are numbers that are generally they're not integers. These are going to be things that have decimal points in them and stuff like that. So, if you're looking at 5 this and 6 that, that's probably counts. But if you're looking at 16.25, that's definitely probably continuous. Ranks, ordinal data. This is what we typically see with pathology. So pathologists are going to say, yeah, you had this many that were category 1, this many category 2, this many category 3. The categories themselves are ranks. Now, you could treat that as counts, because if they're telling you number of animals in a category, you could treat that as counts. But if instead what they're doing is they're grading and each animal gets a grade instead, then you're definitely dealing with ranks. Categorical information is going to be things like this is red, this is blue, this is chartreuse, whatever that color is. Things like yes, no kinds of things. Anytime you're binning things into categories, and sometimes categorical bins are going to end up being counts. It all depends on what it is the question is that you have about the data. The answers to that question right there feeds into this decision on a distributional model. So, in this example, we're going to say, no, we do not have any historical information. Is that realistic? Probably not for this example. For this example, it is more than likely that we do actually have historical information. If you worked in a GLP lab, there's probably historical data. If you worked in a big company, you probably have historical data. If you're an academic, guess what? You probably have access to free data, such as the National Toxicology Program's historical database. Sometimes suppliers of animals also have historical background cancer rates for their animals. So chances are there is historical data out there, unless you're dealing with an obscure species. What type of data do we have? This is counts. Again, we're counting the number of animals that have tumors. And as a result of the fact that these are counts, and these are binary counts, specifically binary counts, and we want to calculate a cancer rate, which is going to be a probability, we'll be looking at using a binomial distribution to represent the counts. The conjugate prior for binomial distributions is going to be the beta distribution. If you don't remember what distributional model we might be using, go pull out that cheat sheet and take a look. I laid out some of the most common distributional models that we use. And again, because this is binary counts, we're talking binomial distribution, and the conjugate prior for that's going to be beta. So The actual cancer rate itself is going to be modeled using a beta distribution. And what's great about a beta distribution, if you remember nothing else about beta distributions, remember this: every value has to be between 0 and 1. So it's beautiful for using for measuring things like cancer rates or measuring things like probabilities. The beta distribution is wonderful for that. So here's our data. We've got vehicle data from this study. 60 animals total. At the end, it was a 2-year cancer bioassay. 5 of the vehicle animals developed tumors. And again, we're gonna count any tumor as a cancer, regardless of the metastatic potential, to be health conservative. This is a very typical thing that's done, for instance, at NTP and US EPA. So 5 of these animals developed tumors, which means 60 minus 5, 55 did not develop tumors. It's kind of interesting, right? So if you wanted to look at this mathematically, this is kind of what it looks like. We have this function with some probability, and we've got these parameters alpha and beta, and that's going to be proportional to this probability with alpha minus 1 as its power. And so alpha here is successes, and the number of successes, I should say. P is going to be your probability. And then 1 minus P is your probability of failure. So P is your probability of success. 1 minus P is probability of failure. Beta minus 1 is the exponent there. Beta is failures. So bringing this together, putting this into a more friendly form where we say explicitly, I'm looking at the beta distribution. It has 2 parameters, alpha and beta. We have 5 successes and 55 failures. This is a beta 5 comma 55 is how we normally write it, which is proportional to the probability, which is the number we're trying to calculate, to the 5 minus 1 times 1 minus P, which is the failure rate, to the 55 minus 1 power. Mathematically, that's what that looks like. Is this really important for us to know? Not really, not unless you need to write this in a manuscript or something for a journal article. That's about the only time you'd ever need to write those kinds of things out. So one of the things we're going to do is we're just going to calculate a random beta distribution. That's what R means. R beta says we're going to randomly draw from a beta distribution. We're going to take 10,000 draws from this distribution. Now you might be wondering, why 10,000? Where did that number come from? I'm doing 10,000 because I like how the histograms look. That's honestly the only reason why. This is just for aesthetics. You want to make sure you get enough draws from the distribution so that you can see what the population looks like. Because at the end of the day, we're drawing inferences about the population. So I'm using 10,000 to make a nice, pretty histogram. If you're to do a number larger than 10,000, it's not going to change how it looks much. Now, if you're to do 5, yeah, it's not going to look this pretty. You need to do a fairly large number, 1,000, 10,000. I don't recommend 1,000,000. That might make your computer go a little bit slow for a while. Here's alpha, 5 minus 1. Here's beta, 55 minus 1. That's what this distribution looks like. Now, you might be asking, but how do I do that in R? Well, here you go. So what I did was I opened up the .r script file so that here I've typed in VEH_plot and this arrow operator. So I'm going to put the histogram into this operator or into this object. And then to show this, I would write VEH_plot. You could just ignore all this and just write hist r beta 10,000 comma 5 comma 55. Just do that. Take off all this other stuff to the left of the assignment arrow. Just do the hist part by itself and run it, and you'll get this. If you do it the way I did it here, you will have to write VEH_plot. Okay, anyway, so there you go. That's what this histogram looks like. Believe it or not, it is the same one that I showed you before. So step number 3, we're going to validate this model. So we look at this, okay. So I've got 555, I got my R beta, 0.08 is the mean for this distribution, and I just happen to color that line blue. And that's what this means up here. So the AB line, V equals 5 over 60. 5 over 60 comes out to about 0.8 or so. COL equals blue means I want to color this line blue, and LWD2 means I want it to be a thickness of 2. So AB line is how we draw vertical lines in plots using the default plotting package. So here, this V is telling it that I want a vertical line, and the equals, this 5 over 60 tells it where I want it. I want it right here, which is 0.08. So we validated the model. The way we validated the model is to say that, yep, that probability is right smack dab in the middle of that distribution, exactly where I'd expect it to be. Yep, this distribution makes pretty good sense. It's not an exact middle, but it's pretty darn close. That's pretty good. So I'd say that this is a pretty reasonable model. And now I can make inferences. And one of the things I'm going to do to make some inferences is we're going to install a package called HD interval. HD interval is a package that allows us to calculate the highest density interval. Now the highest density interval, I'm going to go back a couple slides here. The highest density interval is not the same as a confidence interval. A confidence interval is going to be centered in the distribution. A highest density interval is not necessarily centered in the distribution. It is going to be biased for the largest part of the distribution. So it's going to try to encompass as much of the distribution as possible. In this case, typically we use a 95% highest density interval. So it is going to be the largest expanse that encapsulates the 95% largest part of the distribution. So go back. This is how you install it. You can see I'm installing here, install.packages, HD interval. Don't forget these quotation marks. These are extremely important because that tells R that you're giving it a string, and the name of that string is HD interval. String is what we typically use for words and things like that we want to deal with. Otherwise, R is going to think you're either giving it an object or a command. So if you didn't have these double quotes here and you just wrote install.packages, parentheses, HD interval, parentheses, it's going to assume HD interval is actually an object. And it's not going to treat it like a string, and it's going to tell you, I have no idea what you're talking about. What is this thing? Okay, so it's kind of important that you make sure you've got that. And then to load it, remember, whenever we want to load a package, library, HD interval. Now you're going to say, now wait a second. Here you told me I had to use double quotes, and here you're saying no double quotes. What's going on here? Yeah. So the reason why is because library takes an object, and that object is the name of the package. R knows what all the packages are that you've already installed. And so when you go to load up the package using the library command, it is now looking for the object name that represents that package, which thankfully is the same word that you use to install it. So library HD interval, okay? And the capitalization is important here and here. In both cases, you need to make sure you have things capitalized properly. So the command for using HD interval, as you can probably see right here, I already loaded HD interval. I don't have it up here because I typed it down here in the console. But then here's the command for using HD interval, HDI, and I'm telling it I just want this R beta distribution. So R beta, the random draws from the beta distribution, 10,000 draws, 5 is alpha, beta is 55. I'm not giving it any other parameters, so it's going to actually calculate the 95% highest density interval by default. And you can see here it spans from 0.02 to 0.15. So we're talking, it's basically this region from here to about here. right? Which, if you look at it, is the largest expanse of the distribution. So the question is, what are the most credible values of the probability of cancer for the vehicle given the data? Well, based on the 95% highest density interval, it is between 2.4% and 15.3%. So let me re-explain this. The most credible value of the probability of cancer for the vehicle group is 95% likely to be between the values of 2.4% and 15.3%. So what we're saying is the most likely value is between here and here. So it is between 0.23% or 0.24%, depending on how you round it, 0.24% and 0.15%. It is between this area right here. 95% likely it's in here, based on the information that we gave R. Okay? So based on the fact that we had 5 animals out of 60 developing tumors, there's a 95% chance that the actual probability is between here and here, 0.24 to 0.15. Most likely, it's most likely closest to 0.8, or to be more exact, 0.0789. Okay, which is pretty well right around here, right? So that median is the most likely value, not the mean. The median is the most likely value. It is the point that is smack dab in the middle of the distribution. Again, 0.8. So it's likely close to 7.9%, right? That's what it's most likely close to, because that is the median. So the way we would talk about this is to say, more than likely, the probability of cancer for the vehicle is 7.9%. But, I have some uncertainty here, and my uncertainty is pretty wide. My uncertainty goes from 2.4% to 15.3%. So I am 95% sure it is between 2.4% and 15.3%. But the most likely value is close to 7.9%. That's how we talk about this. That's important because, again, this encapsulates our uncertainty. If we had more animals, would we have less uncertainty? And the answer is yes. And you can do that on your own. But yes, we would have less uncertainty. The distribution would be a lot tighter, which means the highest density interval would be a lot tighter. So now what we're going to do, you just saw how to do this really cool analysis in R using just base R type stuff. I'm going to show you how to do this now in STAN. So we're going to still be using R, but we're going to use something called STAN. And in order to use STAN, first we need to build a STAN file that encapsulates our STAN model. The STAN model is what's actually going to do all of the work. So this is a little bit more intense than what I showed you before with just doing the histograms and the R beta and all that stuff. This is a little bit more intense. But this is really, really, really important because moving forward, we're going to be using STAN almost exclusively for our Bayesian analyses. And the reason why we have to use STAN is because most Bayesian problems are not as simple as the one I just showed you. Calculating something as simple as the probability of a cancer given some exposure is what we call analytically solvable. It means that we can use algebra, and then we can use nice quick and dirty tools in R to come up with an answer very quickly, very easily. Most Bayesian problems cannot be solved that easily. They require a great deal of simulation in order to actually answer the question that we need. So right now, what I'm going to show you is how to do that same thing we just did, that same exact problem, only in Stan. So in Stan, anywhere where you see these two slashes like this, that is a comment. So the Stan interpreter is going to take in this whole entire file, but it's going to ignore any place where these double slashes are. So anything after a double slash, it stops looking at. So the first part of all Stan files is the data section. In the data section, we tell it what we're going to be putting into the Stan model. In this particular case, this is a very simple model. We're going to give it the number of experiments that were done. And here we're saying experiments. The lingo for binomial distributions is kind of different from how we use it in biology. They're saying experiments, we would call it samples, right? So this is actually the number of samples. A binomial distribution represents a number of experiments or tries at something. And so that's the language that is commonly used with binomial distribution. So when it says experiments, when I say experiments on accident, in this particular case, only when I'm dealing with binomial distributions, I actually mean samples. So this part here, int, means that it is an integer. So n is an integer. And this part here, lower equals 0, means that n cannot be negative. The smallest number it can be is 0. Same thing goes for y. Y cannot be negative. Y can only be 0. It is an integer. Y is the number of successes. The model is going to calculate parameters for us. The parameter is going to calculate is called theta. Theta is our probability of success. It is a real number. Its lower bound is 0. Its upper bound is 1. The model that we are ultimately going to fit is a binomial model. The first parameter for the model is n, the number of experiments or samples. Okay, so the number of samples is the first parameter. The second parameter is the probability of success. Y is the number of successes. So what's happening here is we know that we're going to use a binomial model. It's going to simulate values of theta based on the number of samples and based on the number of successes. And the computer is going to search for values of theta where n and y match based on a binomial distribution. We're going to give it a flat uninformative prior, and here is where we have the beta distribution. We're using a beta distribution of 1, 1, which means alpha equals 1, beta equals 1. That is what we call flat or uninformative prior. So let me just show you really quickly what that looks like. So that'll be our beta. I'm going to give 10,000 samples, 1, 1. There you go. You can see how this is virtually flat. It's practically a uniform distribution from 0 to 1. That's what we mean by an uninformative distribution. It is going to give no information to the model as to where theta actually sits. And I'm explicitly telling it I want it to use a beta 1, 1 because honestly, I don't remember from the documentation what is going to use as a prior for binomial. I believe, I'd have to look again, I believe it will actually use a beta 1, 1 by default. But I want to be explicit about this, and this is a good practice, is to be explicit about what your prior is. I'm using a flat uninformative prior. It is going to be beta 1, 1. And so that, again, it's beta, so we know that the value is going to be constrained between 0 and 1. And it's flat, meaning it gives no information. So all values from 0 to 1 are about equally as likely. That's what a flat distribution means. And again, we're calculating theta. So that is what is in the Stan portion of the code. So the way you can make a Stan file is go up here, select Stan file, and you can see, hey, it has a stub for me already. And here it's got a stub for you to do a normal distribution where you want to estimate mu and sigma based on some value some values, I should say, of y. So these would be numbers that you believe are normally distributed, and you want to find out what the mean mu is and what the standard deviation sigma is. This stub right here will do that for you. It's actually pretty nice that they have that right pre-built for you. Anyway, we are going to go into our R notebook, which I will also have this available for you to download to go through. I've written up some background information just in case anybody forgot. Here's the objective that we're going after, was the probability that the vehicle animals develop tumors. Here's how our analysis is going to go. First, I'm going to ask you, if you're to download this, to go look at this stand file and read it and understand it. I documented it pretty well. Then, after you've read it, come back here. Okay, so, okay, read it. So now I come back here. Next, so you looked at the stand file, right? Okay, good. So let's get started. So now what I'm going to do is I'm going to select this line here, library rstand. I'm going to hit Control and Enter, just because I want to run this one line at a time. I could run the whole thing, but I just want to do it line by line. And if I wanted to run the whole thing, I would do Control Shift Enter, and it would run the whole thing for me. But again, I just want to run it line by line. So that this next line, options MC cores equals parallel detect cores. What this means is I want Stan to detect how many cores are in my processor, and I want it to run in parallel. That's what this does. So this makes things go faster because it's going to parallelize the processing and use more of your processor. Rstand options auto write equals true. What this does is, you can see here we're going to read in the Stan model, which was this .stan file we just looked at. What this does is it says save this model after it's compiled so that we don't have to keep compiling it all the time. This is another time saver. So definitely run that line. I like to set a seed every time I'm using R. This set.seed is for R. I use 530727 for a lot of my things. Maybe you can guess what the significance of that is. It has significance to me and several other people, but not everyone will get it. So now I want to compile the stand code. That's a fancy computer science way of saying Stan is going to take this really cool code that we wrote that's extremely human readable, and it's going to turn it into something that's not very human readable, C code. So that's what this is all about. We're going to run this line. I already did this recently, so it went pretty fast for me, you could see. When you do this for the very first time, it's going to take a while. In fact, it may take as many as 30, 40, 50 seconds, depending upon how slow your computer is. So don't worry if it hangs on that line for a while. Just let it go. Get a cup of coffee, get a bag of chips, whatever you got to do, but just let it go. Be like Elsa, let it go. So here's the data line. Here we've got our number of samples. Now you might be saying to yourself, what is this -1 thing? Yeah, let's talk about that. So in Stan, the binomial distribution adds 1 to things. So when it goes to run stuff, it's going to add 1. There's a reason why, theoretically, it does that. I could talk about that all day. The important thing is, know that it does that, subtract 1 here, your life will be simple. If you don't subtract 1 here, you're going to get a slightly different result. It's not necessarily bad, it's just slightly different than what you would expect. So for instance, the mean ends up being closer to, I think, 0.10, or so 10%, versus being closer to 8%. It's not a big deal. It just is what it is. So n is our number of samples. Again, it was 60. We're subtracting 1. Our number of successes is y. That's 5. Again, it's -1. Okay, that's read in. This next part here is actually going to run the Stan model. This is really cool. So the command that we're using is sampling. Vehicle Stan model is right here, this thing right here. That's the first thing we put in. The next thing we give it is our vehicle data. And yes, it needs to be a list. Chains is 3. What this means is this is where that whole parallel processing thing came in. So I'm going to have 3 different processors running the simulation simultaneously. The more chains you have, the better results could be. But generally speaking, you don't need more than 3. I've done as many as 5 before. Typically don't get any better results. Warm up. Okay, so this is what's fun. So it's going to be doing a Monte Carlo simulation, which means it's going to do a bunch of runs. In this particular case, ITER, ITER, is number one it's going to do. It's going to do 15,000 total runs. When the simulation first starts up, a bunch of those early runs are going to be complete and utter garbage because the computer is still trying to figure out where in the world it should even be looking for, in this case, theta. And because we're giving it a flat prior, it really has no idea where theta might be. It knows it's between 0 and 1, so it'll run somewhat fast, but it really has no clue. This is if you run into an issue where your models are taking forever to run and you're using a flat prior, really ask yourself, can I get any kind of historical information? Can I even make an educated guess? Those are all okay, as long as it's educated. Wild guesses, not very helpful, but an educated guess could be very helpful. Even constraining it just a little bit will help. Every little bit will help. So what we're doing is with this warm-up equals to 10,000, what we're telling it is the first 10,000 values that this thing spits out, throw them away. I don't want them. Sometimes we call it burn-in, sometimes we call it warm-up. In STAN we call it warm-up. But just throw away the first 10,000 simulations because I'm pretty sure those aren't going to be cool. To be honest with you, for this particular one, I could probably get away with doing a warm-up of 500 or maybe 1,000 and doing maybe a total of 2,000 iterations. Just saying. Just to be on the safe side, though, because it runs so fast, I don't mind. I'm going to do 10,000 throwaway. 15,000 total iterations leaves me 5,000 that are going to be used to sample and figure out what theta might be. Again, I'm setting a seed for STAN. STAN needs its own seed. It can't use our seed because STAN is actually running in C. So you need to feed STAN its own seed. I forgot to tell you what the purpose of the seed is. This is an important thing. The seed is what makes your calculations reproducible. If you don't set your seed, then what happens is you could get different answers every time you run, which is not a bad thing, but if you're trying to make your work reproducible, it makes it very hard to do that unless you set a seed. So the seed is what is fed into the random number generator that then causes it to spit out numbers in a specific pattern. Because, as you may remember, random number generators are only pseudo-random, right? So anyway, we're going to do that. We're going to run it. Here it goes. This right here means that it's running. There was a stop sign down here, which means it was running, and it's done. It's actually really fast. And now I want to see the results. There we go. So you can see the mean estimate of theta is 0.08, so 8%. The median, which is the 50% mark in the distribution, is also 8%. It's always fun to work when that works out. It doesn't have to. And you can see that the 2.5% goes to 3%, and the 97.5% goes out to 16%. So what I can do is, I think I'll do it down here. I can find the HDI. Bump, bump, bump. Okay, 3 of those. And those are those little tick marks over by the tilde thing next to 1 on my keyboard. And I'm going to see, do I even have HD interval? Nope. So we're going to install it real quick. Install that packages. HD interval. There it is. Sweet. There it is, HD interval. Do that. And then I need to remember it was, you see, I don't always remember. HDI. HDI. See, I try to make it so that you don't have to sit here and watch me type everything, but you know, sometimes I forget. And I want theta. So what this is saying is I want to extract from the vehicle fit object, which is a stan object, theta. And so it's going to be the entire theta, all of it. And it's a lot. Because what I want to do is that those values are the distribution, or the, you know, I could make it into a histogram, but it's the samples of what theta might be, and that's how all these numbers got calculated. And let me just double check. I did this, right? No, I didn't. Ha! I gotta do that first. Now I'll do that. Bingo, bammo. And you can see that our highest density interval goes from about 2.2% to about 14.8%. How does that compare to this over here? This is 2.4 to 15.3. So you know, it's they agree pretty much, right? They're very similar. And so, like I said, that was a 95% highest density interval, and our median was 8%, and our median here was 7.9%. And to be honest with you, Stan is rounding here, so if I really wanted to know what the median was, median, extract, oops, vehicle fit, dollar theta, and you can see the actual median, if you want to take it out, is 7.7% rounded, just close to 7.9%. So there you go. That is how we would do this in Stan. And it is a lot more typing. I'll be honest with you. This is a lot more typing. And I did a lot of documenting here, right? But this is the first step in learning how to use Stan. We're going to build upon this because really I prefer doing all this in Stan anyway. And this is probably the easiest example for us to start with, and then we'll start doing more complicated things going forward in the future. So, have we answered the question that we actually cared about in the beginning? No, we have not answered it yet. Are we going to answer it? Yes, we will answer it. We will get there. Right? But the first thing we had to do was we had to figure out what is the vehicle distribution? What is the vehicle uncertainty and probability, for causing cancer? What is the likelihood that the vehicle is, group is getting cancer? We had to figure that out first. Now that we know that, we can go on to the treatment group, and then we can do the inference that we care about. But first, like I said, the first thing we have to do is we gotta look at the vehicle. And again, what we found in the vehicle is it's about 8%, but I've got some uncertainty on here, right? And so you can see that didn't come out. So let me do this. I'm going to contain everything within the print function so that it will print out those. Although it didn't do it here. Oh, that's unfortunate. There's another way to do that, though. There's always another way. Boom. Okay, so let me do this. That is our uncertainty bounds from 2.2% to 14.8%. And the most likely value is 7.7%. There you go. So we have done this in R, the analytic way. We've done this in Stan now. We've done it for vehicle. Up next, we'll be doing it for the treated group. So hang on, stay tuned. We're making progress. Thanks much.